Okay, I think we can get started. So first of all, hello everyone. Welcome to this session on high-dimensional data analysis. And actually, this specific session, it's a series of sections today and tomorrow on data pre-processing. My name is Andrea Valle. I'm product manager at Lenovo Software. And again, today with me online, we have Greg Veltri that will help me both today and tomorrow. So very briefly, this is the agenda. So we have a lot of stuff to speak about, so I want to go straight. I will show you a few rules of the game in high-dimensional data analysis. I hope they will help you. And then today we will address two topics. I will present two topics. First of all, introduction to pipelines, which is the way in which you will run high-dimensional data analysis experiment in SCS Express. And the second topics that we will present today will be scaling. It's probably the most important pre-processing step. Tomorrow, we will have another session and we will go through the normalization and the downsampling. Why we are dedicating so much time to pre-processing, you will see, because pre-processing, it's basically the way in which you are preparing the data to be analyzed with the popular algorithms, such as tSNE, UMAP, SPADE, FlowSOM, and so on. If you do not perform proper pre-processing, it would be really hard to get meaningful results out of your tSNE, UMAP, and so on. So in this series of two webinars, we will not present any algorithms such as tSNE, UMAP, SPADE, and FlowSOM. You can find several webinars on our website already dedicated to those algorithms. So again, these two webinars are really specific for data preprocessing, which is something that we usually realize it's really underestimated. So the rule of the games. This is probably the most important slide I will show you today. Okay, it's a matter of like manage expectation when you're getting started with high-dimensional data analysis. So those are the 3 points that I want to present you. So first of all, high-dimensional data analysis, it's not magic. It doesn't do any miracles, right? So if you run some high-dimensional data analysis pipeline and you do not get what you want, so you don't get any interesting or cool results, it's either because the high-dimensional data analysis has not been done properly. Maybe you have used bad scaling, or maybe you didn't scale at all. Maybe you use too aggressive downsampling. Maybe you use the wrong algorithms, or maybe the right algorithm, but with wrong setting. Or simply, maybe you have done everything in the right way, but simply there is nothing interesting in the data. So data is data, right? So as any other technique, high-dimensional data analysis, it's an additional tool in your toolbox. It's like imaging, flow cytometry, or doing like Western blot. So you have your scientific question, you have a bunch of tools like flow cytometry, imaging, and you also have high-dimensional data analysis. The second point is, and again, this is really important, when you are sit down in front of the computer running some high-dimensional data analysis, you are the smarter guy in the room. So always be critics when you look at results. Everyone, really everyone, can generate a meaningless, scientifically meaningless tSNE plot or UMAP plot or FlowSOM plot that looks cool and fancy. So you need to practice, you need to take your time to learn how to do it, how to set it, and then always be critics, always validate what you have found with the high-dimensional data analysis with the standard and classical tools like manual gating. Okay, do not blindly trust in what you get out from high-dimensional data analysis. And last but not least, high-dimensional data analysis, again, since it's a technique and it's probably a new technique for you, it requires time and effort to be learned. So I'm sure you want to learn it, you want to master it, exactly as you will never expect to be a super expert in painting or cooking or in any other technique after like one day or even after 1 week. So just do not expect to know everything after 1 day or 1 week. When you get started with high-dimensional data analysis. I'm not discouraging you to learn high-dimensional data analysis. I'm just managing expectations. So be patient, take your time, do things step by step, and try, learn by doing, read papers, send us email, watch video on YouTube, Google, whatever. So do not be impatient and dedicate the time that it requires. And you will see that it's an amazing topic, high-dimensional data analysis. So first things that I want to, first message that I want to convey you is that when you run an high-dimensional data analysis, you're basically running an experiment. It's like running an experiment on your bench. So there are a lot of similarities. Like, we are speaking about flow data, right? So if we think at a flow cytometry experiment, I'm sure you have like a protocol, probably more protocols in your lab. Okay, you don't have just one, because different sample, different scientific question, different antigen, different instrument will require different flavor of a protocol. You can find some general, let's say, action. For example, you need to prepare the sample, and then you need to wash, and then you need to stain, then you need to wash again, then you need to go to the flow cytometer. But the tiny setting, the tiny flavors of each step are really different. And sometimes you also switch different action in different protocol. So which are the different variables that you might encounter when you run a flow cytometry experiment? Well, you might deal with different samples. Okay, it might be like peripheral blood, it might be a tissue, which tissue, might be cells in cultures, which cells, which cultures, and so on. And then you have a sample preparation. This is probably the part that takes more time, right? And the sample preparation, for example, how you isolate cells, okay, single cell suspension, you smash, you lyse, you do some Fico, centrifugation, or whatever. Then you need to stain your cells, okay? And you have different buffer. Maybe you want to permeabilize because you're looking for intracellular cytokine, or maybe you don't want to permeabilize, you want to look at surface antigen. Maybe you want to stain the DNA, different protocol, different temperature, different dilution, different timing. You need to define a panel, okay? So you sit down and use some panel design based on the marker, based on the dye, based on the laser configuration of the instrument. So you see, there is nothing like a unique protocol. And also when it comes to the analytical part, so the very last part, so when you sit down on the flow cytometers, you have all your tubes and you need to acquire them, then you might have like 2 or 3 or 10 different instruments. So you need to select which one. And once you have defined which instrument you want to use, then you need to set it. Okay, set it up like voltages or gains or thresholds and whatever. So you see, every single step has different flavor, okay? And I know you're an expert in these, so just think about how much time and effort you have dedicated to write down this protocol. And the same is happening, and the same will happen if you want to write down a protocol to analyze your sample in with an high-dimensional data analysis pipeline. So it's really similar. We have samples. In this case, those are not cells, but those are numbers, okay? Data file. They can derive from flow or mass cytometer, spectral cytometer, imaging. They can be like cell single sequencing. Once you have those numbers, you need to prepare them to then run the last step, which is the algorithms, right? So what you have in the middle, sample preparation, in high-dimensional data analysis, it's called pre-processing. And again, exactly as it happened on the left of this table, pre-processing, it's not a standardized and unique process like. So it depends on which sample, so which data you are dealing with. It depends on how many parameters you have, how many data you have, which is the scientific questions. So are you looking for a rare population? Are you looking for a very abundant population? So what we are going to do today is learning which are the most used pre-processing steps, Those are just few, like scaling, cleaning, normalization, downsampling. And then we will go into more details about one of them, which is the scaling. And tomorrow we will see normalization and downsampling. Maybe we will organize later on some session on data cleaning also. So once you prepare your data, and this is really important, once your data are ready, then you run the algorithms. So exactly as when you are in your core facility, you need to select which instrument, and you select the instrument based on the, let's say, the power of that instrument. Like you are searching for like 3 laser, 4 laser, spectral, mass cytometry, depends, right? And in this case, you're doing the same. So you want tSNE, you want FlowSOM, you want UMAP. In high-dimensional data analysis, you have the advantage that since everything is done in silico, you can analyze your data as many times as you want, and you can also analyze your data with multiple algorithms. So this is really powerful. But anyway, for each algorithms, you need to sit down and define some settings. So it's not really about running tSNE, okay? It's about pre-processing the data and then running tSNE with some specific setting. And again, for this last part, so tSNE, FlowSOM, UMAP, we have dedicated webinars on our website. So again, today and tomorrow, we will spend time to go through the sample preparation, pre-processing. So this is all formally executed by creating and running something called pipeline. A pipeline, it's nothing else than a series of calculation, series of mathematical steps. So it's exactly what you do with an experiment on the bench. Okay, you have your sample, in this case is the data, and then you do A, B, C, and D. And it's not only a matter of which step you want to run, it's also a matter of which order you want to follow. So if in your protocol you have wash, centrifuge, remove the supernatants, and then resuspend, and then counts, and then sample a given amount of cells, and then stain them. So if you switch those steps, it's not the same, right? And this is also what's happening with a pipeline for high-dimensional data analysis. If you do a scaling and then a normalization and then a downsampling, it's completely different than running a normalization, then a scaling, and then a downsampling. So you also need to be care about the order. And if you attend this webinar and the webinar of tomorrow, you will know what each step does. And this means that you will also know where to put it and in which position to put it in the pipeline. So pipelines usually looks like what you see on the left, which is not really user-friendly. This is what you see in R, in Python, or in vast majority of programming languages. In FCS Express, a pipeline, it's much more user-friendly. You see it on the right. So you see you have different steps, different things that you create, you see here, and you can drag and drop them up and down to change the order. You can switch them on and off by clicking on the checkbox. So what you see here on the right, it's an example of a pipeline created in FCS Express. The pipeline, it's basically a box, a container, okay? And in this container, you have all the steps that will be run from the top to the bottom. So in this pipeline, do not care about what each step really does and says. Just look at the name. Like first step, it's called new flow AI, and then you have new flow rate check downsampling, and then you have new single blah blah blah. Then you have new dynamic. So those are all executed one after the other from the top to the bottom. So the last one is tSNE, for example, and the order matters. How to do this in a FCS Express? So you open your layout. Usually you start by creating some plot and some gate because usually you want to focus on a specific population. You might also run a pipeline on the whole data set. That's fine. But anyway, usually you start by opening at least one plot to look at the data. And then you can go to tools, transformations. And when you open the transformation, what you see, it's this dialog. And when you click on this little plus, you have this list. Now, feel free to go straight to the pipeline. You don't need to care about every other option, actually. Most of the other options are there just for compatibility reasons, because those algorithms such as SPAY, tSNE, PCA, used to be run as a standalone algorithms, not combinable in a pipeline. So this what was happening in the old ages of FCS Express, and actually this is what it's happening in a vast number of other flow software nowadays. But in FCS Express now we have the pipeline, so this is incredibly powerful tool. So just go here, click on pipeline. The pipeline will be created, which is this one. Now it's empty. And once you have a pipeline, which is like an empty box, an empty container, you can click here and start adding steps at your convenience. We have probably like 50, I think, different steps. Don't be afraid. We have organized them in different categories. You see, we have predefined algorithms, FlowAI, FlowSOM, SPADE. We also have Phenograph, downsampling, dimensionality reduction, clustering. So we try to combine them to organize them in a meaningful way. What I want to do now is to show you how to do it in real time with FCS Express. So let me open a layout in which I basically just have few plots and few gates, and then we will create a pipeline. I don't want to create a meaningful pipeline in this stage. I just want to show you how to open the dialog and how to add and remove and drag and drop stuff. So this is my layout. It's a file. This is a file acquired on a Aurora instrument. I have like 3 plots, 3 gates. Again, sometimes you want to run a pipeline on the whole data set. Sometimes you want to narrow down. For example, here I'm manually removing singlets, doublets, dead cells, and I want to focus on lymphocyte. I go to tools. I open the transformation. This is exactly what we were seeing before, and I create a pipeline. This is an empty pipeline, it's just a box, but there is nothing inside. There is no steps, no calculation. We also call this like main pipeline body or first step of the pipe. It's not really the first step, it's just the step 0, let's say, of the pipeline. Root step, you can call it. There are a few options here. The first one, automatically run the pipeline. If this is flagged, everything you change within the pipeline, everything you change within every step of the pipeline will trigger FCS Express to rerun the pipeline. Now, if you have a supercomputer and the pipeline can be recalculated in like a fraction of a second, feel free to keep it flagged. Otherwise, uncheck it so that you can click and run the pipeline only when you want. Then you have gate options and parameter options. Those are the ways in which you define which is the input gate and which are the input parameter for the pipeline itself. Okay, this is just the input for the pipeline. Then in any step, in any point of the pipeline, you will be able to define a subset of parameter on which to run a step and a subset of cells, basically a gate, on which to run a step. So for example, if you want to start with cleaning algorithm, such as FlowAI, and you want the cleaning algorithm to be run on the whole file, then feel free to keep it to no gate. But maybe after the cleaning, you want to narrow down on your lymphocyte. So you can go here and add a gate downsampling step and say, okay, FCS Express, I told you to clean the data using the entire data set, but now, after the cleaning, I want you to focus on the cells gate. And then I can move on and maybe add a scaling step. You see, I can drag it down. I don't want the scaling step to be upstream. I want the scaling step to be downstream. And I want downstream to move on with, let's say, a clustering like k-means. And maybe I want to move on with a tSNE. Okay, you see, I'm adding steps, and the only thing I want to show you is using this drop-down menu to find your step, the step you want, using drag and drop to change the order, and checking or unchecking steps. So for example, maybe you have a downsampling here right after the scaling, and you want to see what happened when you do not run that step, or when you run that step. Okay. Okay, so we have a question. So when you say cleaning the data, what does that mean specifically? Let me go back to that like in few seconds. Thanks for asking. So again, all what I want to show you here, it's how to create the main pipeline and how to add steps. We're not yet to the point of deciding which step to add in which order. We are just to the point in which you know where to grab steps. Okay, so we can delete this. Okay, so let me go back. Exactly as, so there is a question, is there any an easy interface to find out in detail which step, what each step does? Absolutely, yes. Thanks for asking. I can show you these straight away. We have a 1-hour webinar showing and presenting most of the steps we have, but the main and the most complete resource is the manual. You see, just click on manual here. The manual, it's organized in a way that it basically mimics the ribbon bar. So we have a tool, transformations, and under transformation, we have pipelines. So you see here, we have all the pipeline steps. We have hyperlink. And if you go to, for example, PCA, you will be redirected to the PCA. Thanks for asking. Okay, so exactly as we said when we present the comparison between the bench experiment and the in silico experiment, you cannot really find a protocol that works every time, right? But at least you can find a sort of general series of action. So this is probably the ideal sequence of action that you want to perform. So you have your data, you do the pre-processing, you go to the analysis, and then you draw some conclusion, you put together some nice picture. Okay, but which pre-processing step, in which order, which analysis, which setting, that's need to be addressed sample by sample, scientific question by scientific question, and so on. So take your time. We are now on the pre-processing. So here comes the answer to the question, what you mean about data cleaning. So data cleaning is just one of the many pre-processing steps that you might want to perform on your data. So those are just few. Those are probably the most important one, like add metadata, so you can add extra keywords. This is useful if you want to add information that will help you in analyzing your data downstream. For example, you run a tSNE, then you do some clustering, and then you want to compare treated and control. So instead of doing that manually, if you add that information about the experimental group, treated and control, it will be easier. Parameter scaling, this is what we're going to speak in like 5 minutes. Data cleaning, so somehow you are almost all the time, you're already doing some data cleaning by creating gates, right? Exactly as I did in my example. So you draw some gates, remove doublets, you draw some gates, remove dead cells, you draw some gates, remove debris. That's actually, in this list, it's what I mean with population selection. So you do something manually with the gates. Data cleaning in this case is removing bad events that are generated during the acquisition, usually during some fluidic issue, like you have some clog in the instrument, some bubble, you see fluctuation in the intensity. You don't want to spend time analyzing those events, but you don't want to drop away the entire files. So there are algorithms, they can be part of your pipeline, for example, FlowAI and FlowCut, that can eliminate or at least reduce the number of bad events in your data. So thanks for asking. Normalization. Normalization is a way in which you can somehow align your sample or align parameters to make them more comparable. The crucial point is how you want to align them, and we will see this tomorrow. So do you want to align them based on the mean, based on the minimum, based on the maximum? There is not like the best way. there are some tricks, but always do this with care. So you might want to try either with or without normalization, because on some ends you are making parameters and files more homogeneous. On the other hand, you are sometimes flattening the biological variability. Some other time you are introducing biases. So do this with care. Downsampling, we'll see this tomorrow, usually performed to speed up the calculation, but in FCS Express, we have downsampling steps. Those are called density-dependent downsampling. We will see them tomorrow. That may allows you, they're really cool, to enrich rare events, which are usually the events you are interested in. So downsampling, it's not per se a bad stuff, like something that you do only because you want to speed up the calculation, but then you lose rare event. There are tricks to avoid that. So any recommendation on which cleaning algorithms to use, FlowAI or FlowCut? This is a very good question. It's really frequent. The answer is that you need to try. So someone say that FlowAI is too aggressive and that FlowCut it's more gentle. I don't think there is like the best algorithm ever exactly for dimensional height reduction and clustering. So I know you would like to have an answer like always go with this one, but unfortunately, there is no answer like that. So try and see whether one is performing better than the other one. What's the difference between scaling and normalization? Thanks for asking. So scaling, we're going to dedicate the next half an hour. So, and tomorrow we will address normalization. So at the end of those two webinars, you will definitely get this answer. Data merging, this is something that is required by most of the algorithms in high-dimensional data analysis, like dimensional height reduction and clustering, like tSNE, UMAP, FlowSOM, Spades, Phenograph. Usually you need to merge the data. Usually you merge the data at the end of the pre-processing. And this is because you want to run the pre-processing file by file independently. So you want to clean the file independently. You want to scale each file and each parameter independently. You want to maybe normalize each parameter in each file independently. You want to downsample each file independently. Only at that point, when all your file have been cleaned, gated, maybe normalized, downsample, only at that point you will merge those cleaned, gated, downsample, normalized, and gated, blah, blah, blah files. You will get a single merge file, and then that's the file that you will run tSNE and FlowSOM and whatever algorithm you want on. Last but not least, parameter selection and population selection. Those are not specific for pre-processing, because also in the last part of the high-dimensional data analysis workflow, so in the algorithm part, you will have to decide which parameter and which population to use as input data. So the question is, which parameter should I scale? Which parameter should I include in tSNE? Which population should I run my pipeline on? Lymphocytes, CD3, CD4, all cells. So if you want to go more in depth for this, those are really important points. I would suggest watching our main pipeline webinar, which is on our website. So you will find there more details about this, but there is not like a gold standard, okay, gain for parameter selection and population selection. You need to do what you think is the best for your data. And here also you see some additional hints that might be helpful. Okay, so let's go straight. So we have concluded here the first part of this first session. So the one about the pipeline and overall about high-dimensional data analysis with a little bit of introduction of pre-processing. The next half an hour, next 30 minutes will be dedicated to scaling.